All right, now that we have discussed energy and thermodynamics and the transfer of energy, transformation of potential to kinetic, kinetic to potential, let's talk a little bit about those biological catalysts that I mentioned in the previous lecture. And those are the enzymes. Now, enzymes are a class of proteins, and enzymes are used to speed up the rate of a reaction, but again, they don't get consumed during that reaction. And the reason we need these enzymes is because we're looking at these spontaneous reactions that might occur in the cell. So remember, all that spontaneous means in terms of reactions is that the reaction has the ability to move forward without putting any energy into it. We don't have to add energy to the system to make it happen. We might want to add energy of some kind to the system in order to make it happen faster, but that doesn't mean that we have to add the energy to make it happen. So what enzymes do is they lower what's called activation energy. And this is where it gets a little confusing, because even with spontaneous reactions, activation energy is kind of what energy is already there that makes this reaction happen, makes it spontaneous. We don't necessarily mean it's an extra input of energy. But enzymes are these biological catalysts, and they make reactions move without getting consumed in it. And again, so they can be recycled. So let's take a little bit of some time here to take a look at enzymes in particular. So activation energy is required for a non-spontaneous reaction to move forward. So it's the amount of energy that would be required to be put into it. And so that's referred to as activation energy. So initial input to start the reaction. Non-spontaneous reactions, they require us to put energy into it. Spontaneous reactions do not. They don't require additional energy. But there is some energy already within that reaction that's happening, but it doesn't require an additional input. Now, what enzymes do is they lower activation energy required for any reaction to move forward. If it's a spontaneous reaction, it will just make that reaction move forward more quickly. If it's a non-spontaneous reaction, it lowers the amount of energy that's required to make this reaction happen, so the reaction moves forward, and of course, at a greater pace. Now, there are 4 different things that occur during this activation energy. First off, enzymes as catalysts are going to allow molecules to get close enough to cause some kind of rearrangement in their chemical bonds. So just imagine 2 molecules being brought together. We need these 2 reactants to make something happen. We need them to rearrange their chemical bonds so they become a new molecule. And what enzymes do is enzymes create that environment for those 2 molecules to come together. The enzyme is a matchmaker. It brings these 2 molecules together. It also will bring the molecules together and put them in the correct orientation. So we need to make sure that where those chemical bonds, where that rearrangement occurs, is able to meet up with the other molecule where the rearrangement needs to occur. So not only do the 2 molecules need to come together, but they have to face each other in the correct orientation. Sometimes, in order for a reaction to happen, we need a change in pH. Some reactions occur in a very acidic environment. Some may occur in a very alkaline environment. In this case, the area of the enzyme, of the protein that surrounds and actually binds to the reactants, the interior of the protein, when it takes it in, has a change in pH. It may be lined with amino acids that are acidic or lined with amino acids that are alkaline. Either way, it's going to change the pH of just that small little environment inside what's called the active site of the enzyme where the reactants are located. This is going to further weaken the bonds of those reactions in order to get them to break and then rearrange, reform. The last thing that enzymes do is they can stretch or tweak or twist those chemical bonds to weaken them. The more they manipulate those chemical bonds, the weaker those bonds will become, and they'll be able to break and reform new bonds. So this is called a change in transition state, and it's caused by this stretching or tweaking, you could say, of the chemical bonds in the reactants. So activation energy, you'll see these graphs a lot in this class, of course, and in your future classes. And what it's really showing you is here's our Gibbs free energy. Oh, I need to get my pen again. So we have free energy on our y-axis there, right? So here's our free energy. And we have the progress, how far the reaction is moving on our x-axis here. And if we look here, this transition state here, this is the change. So in red up here, this is how much energy, remember, this is our free energy here. So this is the amount of energy that would be required for this particular reaction, these two reactant molecules, to move forward. And this is ATP and glucose. So in order for phosphorylation of glucose to occur, right, this phosphate group has to be broken off and attached. We have to make a chemical bond between this glucose molecule here and this phosphate molecule here. Let's see if I can delete that. And in order to make that happen, there's going to need to be some kind of energy released. So the amount of energy required for that is over here, this transition state. Now remember from the last lecture, it took 3.3 kilocalories to phosphorylate a glucose molecule, and this reaction here released a negative, right, 7.3 kilocalories, leaving us with a change of 4.0 kilocalories. So what this is telling us here is this is our 3.3. We need 3.3 kilocalories for this to happen. And what Gibbs free energy is going to do here, what the enzyme is going to do here is it's going to decrease this to maybe 1.3. I don't know if this number is accurate or not, but I'm just saying it's lowering the amount of energy required for this reaction to move forward. And the way this enzyme is doing it is, remember, this is a protein. We bind something to the protein, it changes shape. So the change in shape might twist or tweak this bond between these two phosphate groups. It may change the OH on the glucose here that's going to get broken off. And by lowering that, we can make this reaction move forward more quickly, and yet we still end up with the same product. We don't change the product. So we have this much, right? 3.3 required without the enzyme, but with the enzyme, it only requires 1.3. Regardless, right? So this is where the reaction is happening. Regardless of which way, we still end up with our change in free energy. Okay. All right. So how do we overcome it? If we need to overcome activation energy, how do we overcome the need for energy to be input into a system? Well, the two most common ways is to add heat, lots of heat, or using some kind of catalyst. In our case, in cells, we use enzymes. So enzymes are the most common way that we lower activation energy in cellular processes. We definitely don't use heat. That cooks things. We don't want to cook things. So we can input energy in the form of heat, but for our cells, we're going to be looking at enzymes. Now, heat can be used to push reactions to a transition state. So think of when you get ill and you get a fever. There are some molecules and signaling molecules and reactions that occur in our cells when we have a fever that occur more quickly because we have a fever. That these enzymes and proteins and these molecules that are part of our immune system actually work better or work faster and have greater efficiency when our body temperature is raised. And that's because their role is in immunology, in the immune system. We also use enzymes to lower activation energy through orientation. Remember, making these two reactants face each other in the correct orientation. And of course, the environment, like pH or hydrophilic or hydrophobic, that sort of thing. So when they are lowering this activation energy, they're straining the bonds. Remember, that's increasing a change in the transition state, the positioning, and of course, change in the local environment. Just repeating those three common ways that enzymes can make reactions move forward and lower activation energy. Now, we need to look at some of the physical features of the enzyme in order to understand what's going on here. So here we have a picture of an enzyme, and we're still using that phosphorylation of glucose step here. And that's because that's the first step in primary metabolism. So on an enzyme, here's the enzyme, this big kind of pinkish reddish blob here. Here's our enzyme. And where our two molecules actually fit or bind to the enzyme is referred to as the active site. These two molecules here, the ATP and the glucose in this example, are these substrates. These are what actually will bind the reactants that will bind to an enzyme for a reaction to move forward. So we have our substrates right here, ATP and glucose are the substrates. Here is our enzyme. And when they come together, they form an enzyme substrate complex. So you can see when these guys bound, here's our ATP molecule here, and here's our glucose here. When these bind to it, our enzyme here has changed shape. The change in shape has caused the phosphorylation of glucose. When this chemical change, when this energy change or this rearrangement, I should say, when this rearrangement occurs, this causes a secondary change in shape. So look at how much further apart, right? We have a bigger gap here. When this enzyme opens back up, we now have a release of our products, our product being ADP and glucose 1-phosphate. Now our enzyme goes back to its original state. And so the enzyme now is ready to take on new substrates and repeat this process. And so this just is going to keep occurring over and over and over. Now, when a substrate binds to an enzyme, keep in mind that enzymes are very, very specific. They have a very high affinity, which is also referred to as specificity for their substrate. And the example that's used is a lock and key mechanism. Now, the key to my front door is not going to fit your front door, and the key to your front door will not fit my front door. Just like neither of our keys will fit my neighbor's front door, and none of us have keys to the door to the college, to the lab room that we use. So each of the doors in this particular analogy is the enzyme, and the keys are the substrate. You have to match the substrate to the right enzyme. So these enzymes have very high affinity as well as high specificity for their substrate. An induced fit is when a substrate binds to an enzyme and the enzyme actually changes shape. That's what is referred to as an induced fit. A prosthetic group is when an enzyme has an additional molecule attached to it, usually permanently, in order for that enzyme to carry out a reaction. An example of this would be iron bound to hemoglobin. So there are two different types of prosthetic groups that occur on enzymes. The first are cofactors. So cofactors are inorganic ions, such as the iron bound to hemoglobin. A coenzyme would be an organic molecule that might participate in a reaction, but it's just like the enzyme, it'll be left unchanged after the reaction. So our B vitamin video is not working, so I am going to leave this for you guys to watch outside. Now, enzymes are very specific, but they also need very unique, or not unique, but they need very specific environments in which to work. They are proteins, so they can be denatured, and they can be denatured fairly easily. If you think about our bodies, the internal workings of our body, we're a constant temperature, we're a constant pH. Those two factors right there are very important in the functioning of enzymes. Most enzymes have a maximum rate of function in a very narrow range of temperature and pH. For our case, most of our enzymes function best or optimal, what they call optimal conditions, are 98.6 degrees Fahrenheit or 36 degrees Celsius body temperature and a pH of near neutral, about 7.2. When we get outside of this range, the further out of that range you get, the less function or the slower or less efficient the enzyme function will be. when we get really far out of optimal conditions, then enzymes don't function at all. So this would be an extreme change in pH or an extreme change in temperature. You may have observed this in lab, in your enzyme lab with the potato enzyme. Enzymes can be denatured by changes in temperature, change in pH, and there are some chemical denaturation properties too. There are some compounds that may be able to denature enzymes, such as a pharmaceutical or a drug, something like that. The problem with denaturation of enzymes is that it is usually permanent. Not always, but most of the time denaturation is permanent and the enzyme becomes completely non-functional forever. If this were to happen inside of cells, the enzyme would eventually be broken down and preferably replaced. But one thing that we have not talked about here is the regulation of enzymes. So enzymes are not only can be denatured, but we sometimes need to turn off an enzyme or turn an enzyme on. So enzymes do need to be regulated, and we'll look at some properties of regulation when we talk about metabolism.